Francesca Tuazon. Francesca is going to be attending in the fall the PhD program in the CMD program at the University of Pennsylvania. And her talk is role of microRNA processing in cellular differentiation and reprogramming.
My name is Francesca Tuazon, and I'll be talking about the research that I've been doing in Mike Wormington's lab for the past year that looks at the role of microRNA processing in cellular differentiation and reprogramming. So microRNAs are small, non-coding RNAs that are involved in the post-transcriptional regulation of gene expression. And they do this by directing either the translational repression or degradation of their target messenger RNAs. MicroRNAs are synthesized in two basic cleavage steps. The first happens in the nucleus, and the second in the cytoplasm. And it's the second cytoplasmic cleavage that creates the mature microRNA. And that forms, it assembles to form the RNA-induced silencing complex, or RISC. And that mature microRNA directs the activity of RISC to either translationally repress or degrade the target messenger RNA. So as most of you probably know, microRNAs are emerging as crucial players in various disease developments or just regular cellular function. But most of these studies are correlative, linking a certain expression profile to a various disease or cellular development. And what my research focuses on is teasing out whether those expression profiles are the cause or the downstream effect of those observed cellular processes or disease developments. And I do this by targeting the processing proteins involved in microRNA biogenesis. And by targeting these different proteins, like the ones that I focus on are DROSHA, DGCR8, Dicer, and AGO2. By targeting these different proteins, I'm able to directly look at the role of microRNAs and their functional role in these different disease developments. And these three up here are processing enzymes that are involved in the cleavage of microRNAs. And then AGO2 is a protein that, along with RISC, helps determine the actual microRNA's function. So I look at microRNA function in two developmental contexts, in differentiation and reprogramming. The model that I use for differentiation is human neuroblastoma because that has a specific cancer stem cell that can differentiate. And then my model for reprogramming is the Xenopus stage 6 oocyte, which I'll get into a little bit later. So first to talk about the role of microRNAs in differentiation, I'll introduce my model, which is, as I mentioned before, the neuroblastoma. And neuroblastoma is a great model to study differentiation in because it's a cancer that's distinguished by three different cell types. And the cell type that I focus on is the I cell, or intermediate cell. And that's because it's a stem cell that has the capability to differentiate into either of these two other cell types, neuronal N cells or S cells, which have a more strong glial phenotype. And what's really great for me is that this can be induced chemically in the lab. So I can combine this system along with inactivation of microRNA processing proteins and ask a directed question of if microRNA processing is necessary in I cell differentiation. So previous work in the Wormington lab had characterized I cell differentiation. And what of particular interest to me was the use of BIMI1, which is a neuronal marker. What Alex found was that BIMI1 expression increases in N cells and decreases in S cells relative to the I cells. So this gives me a marker that I can use to track I cell differentiation. I followed up on this by performing a time course of I to N cell differentiation with retinoic acid. And what I found was that BIMI1 levels increase around days 9 through 12, which indicates that eye cells are undergoing that neuronal differentiation. And what's important to notice is that at that same time, Dicer and Drosha levels are decreasing, but then what's of particular importance is that DG is constant throughout the whole process down. So knocking down GGCR8, not only because since it's there constantly, it might be pointing that microRNAs are involved in this whole process, but also interestingly enough, DGCR8 is the only processing protein that is only involved in microRNA processing. For example, Drosha is also involved in ribosomal RNA splicing, and so is, and then Dicer is also in siRNA cleavage. And so in order to knock down DGCR8, I use short hairpin RNA constructs, which inhibit translation and destabilize the target messenger RNA. And there were 4 constructs available in my lab that target different sections of the DGCR8 gene, both in the coding and in the non-coding region. And so I performed some preliminary transfections to see which construct was the most effective. And I had to do this in a sarcoma line instead of the neuroblastoma line that I'm really interested in, because the eye cells are really extremely difficult to transfect. So I wanted to first do just a diagnostic type of transfection. And what I found was that constructs 2 and 4 decreased DGCR8 expression. And this is my Western, this is another from a different lab, a graduate student who's doing similar work with the same constructs, and his Western just shows it a little bit more clearly than mine. I also wanted to follow up and see what was happening to the DGCR8 message. And so I did this through qPCR and looking at the levels of DGCR8 messenger RNA. And I saw that construct 4 has the greatest ability to knock down DGCR8, which confirms what I had seen in my Western. So the constant levels of DGCR8 make it a good target. And then I've shown that I'm able to knock down DGCR8 in a preliminary cell line. So the next step is to do the transfection in my desired BE2C eye cells. And then repeat the time course and see how that changes the ability of these eye cells to differentiate. Unfortunately, though, because of the technical limitations that I mentioned before and the limited time that I have at UVA, I decided to switch directions just because it would have taken a lot of time to actually transfect those BE2C cells. So I changed my direction and my focus to looking at microRNAs and reprogramming. And as I mentioned before, the model that I'm using is the stage 6 oocyte. And the reason that I'm using this particular model, the question that I always get, is because these oocytes have been shown through the 60s that they're able to reprogram differentiated nuclei back to a stem cell-like state. And that's because they're transcriptionally and translationally active. And that kind of makes sense when you think of an egg and a sperm coming together and through fertilization, then creating this zygote that's made of completely embryonic stem cells. So an overview of the reprogramming experiment is with the nuclear transfer, taking the nuclei that you're interested in that are differentiated, transferring them into the nucleus of your oocyte, letting them incubate, and then you can see the reactivation of embryonic stem cell marker genes, and the ones that I'm looking at are OCT4 and NANOG2. Also in my experiment, the donor nuclei that I'm using are the 2FTGH cells because they do not express any embryonic stem cell markers. And in order to look at microRNAs in particular, I can knock down those processing proteins I've mentioned before in the oocyte to ask directly if reprogramming requires microRNA processing. This whole nuclear transfer, though, had never been done in my lab. And so first I had to show that reprogramming was even happening. So what I did is I looked at the levels of RNA in the injected 2FTGH nuclei after just a regular nuclear transfer in an intact oocyte. And what I saw is a time-dependent increase of the embryonic stem cell markers that I had mentioned before, And this is also confirmed when I do gene-specific RT-PCR beforehand to cut through any oocyte background, just consistently showing that reprogramming of the somatic nuclei is happening. So after showing that the normal process can happen, I had to be sure that I could mess up the process. So I needed to knock down the microRNA processing proteins in the oocytes. And there were two available methods to me in my lab. The first was the use of an antisense oligonucleotide, which causes the degradation of the different target message, and then also of morpholinos, which block the translation of the target messenger RNA. So it was necessary for me to see which was more effective for different processing proteins in the oocyte. And I did this for all of the processing proteins that I've mentioned before, and the one that I saw the most consistent results with was with dicer. And so looking at the AONs versus the morpholinos, I saw that with morpholino injection, dicer expression is decreased at 72 hours, and then also at 48 and 72 hours with the AON injection. So where I am right now is I've shown that the normal reprogramming process can happen and that I can manipulate the microRNA processing in the oocyte. So the next logical step is to combine those together and to perform a nuclear transfer in an oocyte that's lacking microRNA processing to directly see if microRNA is required for that.